Hi everyone, I am Shutong Zhou. In this video, I am going to discuss the exploration of blood-brain barrier on the chip and its application. So the blood-brain barrier refers to the microvasculature of the central nervous system. In this picture, the blood capillary is in the middle in pink color, and there are multiple kinds of cells surrounding it, acting as a barrier. This structure is very important because no brain cell is further than about 25 micrometers from a capillary, so it is the main obstruction for drug delivery. Other reasons why blood-brain barrier is important include its disruption is presented in various neurological disorders. For example, a study in 2019 pointed out that Parkinson's disease may involve the infection of a specific bacteria species across the blood-brain barrier. Also, another study revealed that similar pattern of BBB disruption is seen in multiple disorders, which may contribute to the discovery of a core BBB dysfunction module. So the disruption of this barrier may be considered as one of the convergent symptoms of different diseases. Drugs that target the BBB may have the effect of controlling symptoms if this area can be well studied. In terms of drug delivery, it is important to make sure the drugs for CNS can firstly cross the barrier into the brain. There was a measure of the ability of molecules to cross BBB named the blood-brain barrier score, but if the drug is needed in large amounts, it is not efficient to only deliver the drug via diffusion. Instead, there are active mechanisms involving proteins in the membrane which take things in by changing their 3D structures, for example, the process of receptor-mediated transcytosis. While patient studies are obviously not suitable for testing which involve large number of trials, the technology of BBB on a chip provides a reliable and practical way for research. So this is one of the early versions of the chip. They called it a 3-lane organoplate in which endothelial cells were co-cultured with pericytes and astrocytes. The purpose of this layout is to assess the barrier property of endothelial cells separately with pericytes and astrocytes. The results showed that both cell populations have the ability to prevent certain molecules crossing the barrier. And this is a more well-known appearance. The core of this chip is to recreate the blood-brain barrier's ability to control the substances crossing either from the blood side to the brain side or the opposite direction. The studies of drug delivery mainly focus on routes entering the brain, but the other direction can also be important. And to assess the permeability of blood-brain barrier, different antibodies can be applied. For instance, in this study of Alzheimer's disease, the wild-type cells fully prevented the diffusion of a specific antibody across the barrier, while the ones with Alzheimer's disease had this leakage, which resulted in the presence of antibodies in the brain side, which can be illustrated in the scan. With these approaches of blood-brain barrier chips, one of the most important questions is it easy to establish the environment in the body? For example, one of the studies used cells taken from human samples and genetically modified them to make them immortal, but that would probably alter some other properties of the cell. Other ones used induced pluripotent stem cells, which should be more similar to the cells in the physiological environment. Another factor is the layout of different compartments. One researcher in 2020 designed a 3D layout with vascular space of the brain microvasculature in the upper layer, and pericytes underneath the membrane and astrocytes in the lower layer. It was discovered that cells reacted differently in the 3D layout by downregulating a specific astrocytic protein. The researchers concluded that the 3D layout was more physiologically relevant because it was able to capture more properties of the actual human body environment. And here's another example. This study in 2018 created a different mimicry. They built this linked model separating the influx across BBB, parenchymal compartment, and efflux across BBB, and found that the endothelial cells and the neurotransmitter synthesis system in the brain can have a bidirectional relationship. This posed another question regarding whether the chip can fully replicate the physiological environment. It is still not clear whether we should include as many factors as we can because the brain is highly connected and the details of connectivity remains a mystery for us. Keep adding factors may result in a much larger chip, which complicates the aim of this blood-brain barrier on a chip technology. Therefore, the development of blood-brain barrier on a chip is still in its infancy period and requires more exploration. And now let's move on to the application of blood-brain barrier chips. It is a very good tool for disease modeling as we can not only mimic the healthy environment but also the environment under the conditions of different diseases. There have been approaches to use blood-brain barrier chips to study Huntington's disease, Parkinson's disease, and Alzheimer's disease. The main approach can depend on the disorder itself, but for example, in the case of Huntington's disease, symptoms include deficiency of the MCT8 gene, which affects the blood-brain barrier and causes decreased permeability of T3, which is a thyroid precursor from blood to the brain. In the study that induced a MCT8 mutation by CRISPR and verified this change in blood-brain barrier permeability for T3. Therefore, the chip can be used to verify the effects of disorders on the blood-brain barrier. Of course, this may be only one of the factors. The results of this kind of study can infer future directions for disease research. Another important use of blood-brain barrier chips is potential central nervous system drug testing. As I earlier mentioned, there can be dedicated routes for certain drugs to enter the brain. The chips can be used to study the active processes of the membrane. It is also possible to combine disease modeling and drug testing for more complete study in neurological disorders. Also, in terms of medication, there is a possibility to establish a chip for different individuals which capture the properties that may vary between patients, so that they can be used to test treatments that are specific for individuals. However, these hypotheses still need a large amount of testing and verification before being used in medication. The problems I discussed earlier still require consideration. Thus, in conclusion, blood-brain barrier on a chip is a very new and exciting field. If the problems can be overcome, it can bring huge advantages for multiple areas of study. So, thank you very much for listening.